from the previous class to this new class. In the previous class, I talked about history of genetics, what Gregor Mendel did. What Gregor Mendel did. He found factor, he called it factor, that causes phenotypic variation in pea plant. He found Mendel's law, principle of law of inheritance, law of segregation, and law of independent assortment. That in the law of segregation, during the formation of an individual, only n haploid number of chromosomes take part in the formation of an individual. And law of independent assortment says that during the formation of individual, the gametes, the n haploid number of chromosomes, n haploid chromosomes are sort randomly, are independent of each other. And thereafter, I told why Mendel's work was very successful. Mendel was very successful because he was a fantastic bookkeeper. He learned from the mistakes of other previous researchers. He is a pollination of the pea plant, he is a fertilization. And from there, I speak to environment by gene interaction. The gene, we know how gene interact to create expression in individuals, but gene doesn't do it alone with assistance of environment. So environment modulates the degree of gene expression. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? I have no question here. So let me just continue. So then gene with the component of genotype. When two genes come together, they form genotype. Combination of genes form genotype. Genetic makeup, genetic constitution. Gene from father, gene from mother come together to create a new genetic makeup in the offsprings. So this gene react differently in environment. That's why phenotypic expression is a combination of genotypic variation and environmental variation. An environmental variation is a variation due to an environment. And everything outside control of gene is environment. Genotypic variation is variation in population due to genetic makeup. And we have variance of component. For instance, genotypic variation is modulated by the comprises of epistatic dominant variation, additive variation, and epistatic variation or variant. Dominant variance, epistatic variant, additive variant. These are the three types of variants that is influencing genotypic variance. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? So, having said that, we left heritability because this genotype, this gene that is coming from the parent to the offspring, environment influence the degree of expression. And ability of gene to move from one generation to another is what called it has to do with heritability and heredity. Movement of a character of a gene from one generation to another is called heredity. And when we say a gene is hereditary, means that the gene can move. That gene can move from one generation to another. Then we say that gene is hereditary. Am I speaking sense? Am I speaking sense? And having said that, we move to heritability. Heritability too has to do with the degree to which the offspring inherit, or extent to which degree to which the gene move, transmit itself from the parent to the offspring. Now the degree, not just moving alone. What is the degree? I mean degree of expression. The degree of expression. So it ranges from 0 to 100. 0 to 100 or 0.1 to 1. The degree. Am I speaking sense? Am I speaking sense? Am I speaking sense? I'm saying Yaya, I'm saying Joel, I'm saying Ephraim. Fantastic. Fantastic. So that heritability now determines the degree of expression. Gene, a character that has low heritability, you need to use the environment to improve the performance of such character. A character that has higher heritability, you don't need selection, because it will always move itself from the parent to the offspring, and it will express itself very well because of higher heritability. Am I speaking sense? Am I speaking sense? Having said that, so that took us to heritability in broad sense, heritability in narrow sense. Heritability in broad sense is governed by the entire genetic makeup. That's why it says in broad sense, degree of genetic examination. Where heritability in narrow sense is governed by additive gene action. So that the gene coming from the parent move from the parent into the offspring jointedly, in unison, equally, creating a co-dominant action in the offspring. Both genes move from the parent onto the offspring, express themselves equally. They express themselves equally. They express themselves equally. That's why it's called it measures the degree of genetic, it measures the degree of resemblance. It measures the degree of resemblance in the offspring. That's what made the parent the children look very much like their parent. So, and what causes that? Additive gene action, which causes it, gives it ability to breathe through. PRUE, ability to breathe through. The recorded class I sent to you. How many of you, if you have seen that recorded version, thumbs up, let me see. If you have seen if you have equally seen the summary, I've seen this summary and detail of the transcript of what I taught. Thumbs up, let me see. Fantastic. So co-dominant form of gene action is manifested in additive gene action. When the two characters express themselves equally. So, as I said, that we have other form of gene action. We have other form of gene action. Like epistatic gene action. I've briefly explained. Crossover and gene linkage. In gene linkage, gene on the same chromosome. Crossing over, when two adjacent chromosomes exchange chromosomal part. But now, two adjacent chromosomes that lie side by side, exchange chromosomal part. The exchange chromosomal part, ordinarily, they're supposed to segregate. They follow during gametogenesis, they segregate. They're supposed to segregate. They're supposed to segregate to give us an haploid chromosome that will take part in the independent assortment. But instead of them to segregate, they exchange chromosomal parts, and it causes genetic diversity. I'm saying Joel, as I'm saying Joel. Then what Joel? Which question are you trying to ask question? Let me see if Joel is asking question. Joel, are you trying to ask questions? I see you raising your hands. So, having said that, they refuse. So they exchange chromosomal parts, and they may eventually segregate in crossing over. But in gene linkage, when gene on the same sister chromatid, refuse to segregate. They refuse to segregate, and they move together onto the hospitals. We have, for instance, a character for tallness, for tallness from the parent, and a character shortness from the two parents, from the mother, tallness from the father. The gene capital TT suppose residing on two homologous chromosomes, and T small t residing on two homologous chromosomes. Instead the capital T to segregate into T gamete, 2T gamete, small t gamete in the mother, the two genes move directly, they move together onto the offspring. That's the gene linkage. So we have two examples of gene linkage. We have coupling and repulsion. Coupling and repulsion. The two examples of gene linkage. Coupling and repulsion are the two examples of gene linkage. Coupling is when like I said, coupling is example of gene linkage. What do I say? Coupling is an example of gene linkage. And we have two types of gene linkage. We have coupling and we have coupling and we have repulsion. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Fantastic. So in coupling, two recessive genes got linked together and are inherited together. Am I speaking sense? It's like the network is low. Am I speaking sense? In coupling, when a dominant gene and recessive gene move together, when the gene on the same homologous chromosome, two pairs of homologous chromosomes supposed to segregate, they don't segregate. They move together into the offspring. They are inherited together in the offspring. It's called coupling. So when two recessive pairs of genes are inherited together, we call that one is called repulsion. When two dominant genes move together, when two dominant genes are inherited together, or two recessive genes are inherited together, called it repulsion. But when a dominant gene and a recessive gene, or let me say a dominant allele, let me put it that form. When a dominant allele, can I put it in this way? Let me put it this way. A repulsion can be simply defined as when a dominant allele stick together with another recessive allele. When a dominant allele stick together with another recessive allele, that is called coupling. Meaning that a dominant allele or the two chromosome pair and recessive allele are inherited together. But why repulsion? Repulsion is when two recessive allele are inherited together, or two dominant allele are inherited together. That's repulsion. There are two types of linkage. So linkage disobey simple Mendelian law of inheritance. Epistasis disobey simple law of Mendelian inheritance. They disobey segregation. Am I speaking sense? So you may be asked, what are the two types of gene linkage? What is crossing over? When two adjacent chromosomes crosses each other and they take chromosomal part, that point of exchange is called what? Chiasma. When we have multiple points of exchange, multiple points of manifestation of exchange is called chiasmata. We have two types of linkage, crossing repulsion and the coupling, repulsion. So when coupling occurs between a dominant allele and recessive allele, coupling, dominant allele, etc., they stick together. But repulsion occurs between two recessive allele or two dominant allele. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? So crossing over occurs between two adjacent chromosomes, while gene linkage occurs between two sister chromatids, two homologous chromosome pair. So now we'll move to, let me turn to, we have another one we call epigenetics. Epigenetics is another form of gene action that disobey simple Mendelian inheritance, like methylation, gene methylation. When a gene is methylated, epigenetics doesn't change DNA sequence, but it's an attachment of certain compounds that influence gene expression, like methylation of thymine at carbon 5 position. When thymine is methylated at carbon 5 position, is a form of epigenetics. Another one is ubiquitination. That ubiquitin gene used to influence post-translational expression, meaning that after DNA is transcribed to RNA sequence, mRNA sequence, ubiquitin gene can attach itself to DNA sequence and improve or lower gene expression. So this epigenetics, these are extraterrestrial factors that influence gene expression without actually disrupting the DNA sequence. Am I speaking sense? Am I speaking sense? So epistasis is one of them. Another one is epistasis. And what is epistasis? Epistasis is when a gene at one locus, get it very clearly, when a gene at one locus masks expression of another gene at other locus. So it's an interloci, interaction. Am I speaking sense? Am I speaking sense? So it is interloci gene interaction. That's why I call it interallelic interaction. A gene at one locus marks expression of another gene at other locus. So it's an interloci between two loci. The location a gene is residing on the DNA is called locus. Singular. When it is plural, you call it loci. So, but the gene action between the same locus is called intralocus. That between dominant gene action and recessive gene action. And I have extensively explained that. Am I speaking sense? Am I speaking sense? Fantastic, fantastic, fantastic. Now I move to epistasis. Epistasis, when a gene at one locus masks, suppresses the expression of another gene at other locus. A gene in Latvia can mask expression of another gene in Akwanga, interlocus. So these disobey simple Mendelian principles. Epigenetics, crossing over, gene linkage, epistasis, they disobey simple Mendelian law of inheritance. They disobey law of segregation, they disobey law of independent assortment. So there are different forms through which gene can move from the parent to the offspring without following simple Mendelian inheritance. And largely, characters follow simple, especially simple characters. They follow simple Mendelian inheritance. Am I speaking sense? So that is why dominant gene action is a kind of intra-allelic gene action interaction. While epistatic gene action is inter-allelic interaction. Epistatic means to mask. One gene and one locus mark the expression of other gene at other locus. Am I speaking sense? Am I speaking sense? Am I speaking sense? If I'm speaking sense, thumbs up. If I'm not speaking sense, just thumb down. Then I know I'm not speaking sense. That's fine. That's fine. So, having said that, we get to different types of epistatic gene action. Different types of epistatic gene action. Number one. So let me now say epistatic gene action, crossing over, gene linkage, create a striking deviation from simple Mendelian ratio. They create a striking deviation from simple Mendelian ratio. For instance, I can therefore say, let me say, I can take classical Mendelian ratio, for instance, in the hybrid crossing, for maybe wrinkle round and wrinkle round, when you have a round yellow mango or a wrinkle green mango, wrinkle one character, Green, another character, color and shape. Dihybrid crossing. So in a classical Mendelian ratio, for instance, dihybrid crossing, phenotypic ratio is 9 to 3 to 3 to 1. It does not occur. There's a striking deviation from this in epistasis, because characters are influenced by more genes. So there is striking deviation from simple Mendelian ratio, Mendelian inheritance, phenotypic ratio. For instance, we have recessive types of epistasis. We have recessive epistasis. We have duplicate epistasis. We have duplicate. We have recessive. We have complementary epistasis. Am I speaking sense? Am I speaking sense? But let me speak to recessive epistasis. In a recessive epistasis, we can see a phenotype that has been modulated or being mediated by recessive epistasis in mouse, in just our common house mouse, where color codes exist, which is agouti color code. Agouti color code, like zebra-like color, white and black. That's agouti color code. A-G-O-U-T-I. Agouti color code. So the mouse, the agouti color does not express itself due to simple Mendelian inheritance, but due to recessive epistasis, which is epistatic gene action. So the wild type, meaning that the natural one, the wild type, the one that has not suffered variation, suffered evolution. I mean, no, the one that has not suffered mutation. The wild type is agouti. The hair is like white and black. Most part with narrow, we talk narrow or yellow band. Talk about white band. Talk about white band. Come about the yellow band. I mean, white and black band with both part black with narrow yellow band near the tail. But agouti is more or less like black and white. So the one that suffered variation or mutation become albino. Without it, the one that suffered mutation, you become albino. Or variation, you become albino. You become albino. So this type of house mouse, most musculus, that is the name, zoological name, most musculus, has a agouti color. But the next variation to it is albino. If it's not good, it will be albino. So an albino always break through. So, but how do we get this black color? When the black color, when the yellow pigment in agouti, yellow pigment in agouti is black color. The black is recessive to agouti. That black is just agouti. So when pure black mice are crossed with albino, when black mice are crossed with albino, the progeny will all be agouti. When black mice are crossed with albino, the offspring, the progeny, will all be agouti. But when F1 crossing within each other, the black and albino appears in ratio of 9:3 to 4. So when you cross black mice and albino together, epistatic gene action will not create 9:3 to 1 ratio. It will not. It will create 9:3 to 4 ratio in the progeny. This striking deviation from classical Mendelian ratio 9:3 to 3:1 is called by recessive gene action, recessive epistasis. So if you do a simple, if you do a simple crossover, if you do a simple crossover, a simple crossing between black albino and within a simple crossing between albino and black rats, you will have 9 to 3 to 4 instead of 9 to 3 to 3 to 1. Am I speaking sense? Am I speaking sense? That is fine, fantastic. So what are the conditions? Mean the conditions that will make you to have 9 to 3, meaning that when you say 9 to 3 to 4, 9 agouti black with some white patches. We have 9 agouti white with some black patches. That's agouti color, like gray, black. You have 9 agouti, 3 black mice, and 4 albino. And how does it happen? That is the phenotypic ratio, 9 to 3 to 4. Now, for you to have black, for you to have agouti, remember we are talking about recessive epistasis. When it is recessive, it will give you albino. When at the 2 locus, if the, let us say, at 2 locus, black color and agouti, and albino, black color and albino. So at the gap, when you will have albino when the C alleles and A allele, the 2 C allele and the 2 A alleles are recessive. You will have albino. You have albino too when the 2 C allele and 1 A allele is dominant. Anywhere you see 2 C allele, you know, like I told you, you are crossing between black mice and which is black color C alleles and albino color A alleles. And the black color is always dominant. But anywhere you see the C allele, which is coding for black, in recessive, anywhere it's recessive, it will create albino. It will create albino mice. Anywhere you see double recessive, anywhere you see double recessive C, it will create albino. So it didn't follow simple Mendelian inheritance. It didn't follow simple Mendelian inheritance. So you have, so that is what creates albino in agouti when you cross agouti mice and black mice. Another recessive. So, like I told, let me say something to you. We have black color C and a CC and agouti and albino. CC and albino. Let me quickly explain. CC and CC is coding for black color. Black color. So anyway, mean that CC with which is dominant black, and AA. Don't forget, we are talking about mono dihybrid crossing. So CC black is dominant over AA albino. So C is strong C is shown, the black color is shown. When you now cross it, it's albino. Albino is small letter C and small letter C and capital letter AA. Albino. AA is for albino. But for albino, for the rat to be albino, the two allele for black is recessive. Then when you now cross it together, you now realize that anywhere you see double recessive CC, double recessive color of black, when black is in a double recessive form, it will give you albino. And when you do the Punnett square crossing of this, you will find 3 albino, which is CC, small letter C, 2 recessive C, and 2 dominant A, albino. Again, 2 recessive A, 2 recessive C, and a dominant A and recessive A, with albino. You see in another place, double recessive C. So you see 1, 2, 3, you will see three observations where you have double recessive, about four double recessive condition of C, you give you albino. And black, and you see agouti. Where you will see agouti is anywhere you see between the two loci of C and A. When C is in the dominant condition, A is in dominant condition, you see agouti. At locus CC, at locus AA. And if you see C in dominant condition, A in dominant condition, you will see agouti. Am I speaking sense? Am I speaking sense? Am I that fantastic? Fantastic. So you see black. You can only see black when A is the recessive condition. You only see black when A, and you can find that one only in 3 places. When I give you the materials, it will broaden your understanding more. So we'll go to dominant recessive. I've dealt with recessive epistasis. Recessive epistasis. I'm going to dominant epistasis. In dominant epistasis, we are talking, example is shown in color, in color of sheep, color pattern of fleece. The fleece, the ear of sheep is called fleece. The color pattern of fleece in sheep, we have white, which can be white and black. White is the gene W for color for white, while B is the gene for black. Dominant epistasis. In this kind of epistasis, there is a striking deviation from simple Mendelian principle, a Mendelian ratio of 9 to 3 to 3 to 1, but here you see 12 to 3 to 1. So this dominant epistasis will give you 12 ratio, 12 to 3 to 1, meaning that you see when you cross white sheep and black sheep, you will have white sheep 12. You will have black 3. You will not see brown 1. And let me explain to you how you come about this. Let me explain to you how you come about this. So for you to see anywhere you will see white, the white color, the 2 alleles for white color will be in dominant condition. Will be any of the 2. That white color, which is coded for by W. So anywhere where the two alleles for white, WW, any one of them is in dominant condition, it will give you white. Any one of them is in dominant condition, it will give you white. In recessive epistasis, where you see in recessive epistasis, and this opposite of recessive epistasis, the color of agouti color, and albino color in rats. That is mouse or mice. Now, so anywhere you see the albino color. When you see black color in double recessive condition, anywhere you see black color, which is C in double recessive condition, the 2 in double recessive condition, it will give you albino. It will give you albino. Now, in dominant epistasis, the white color has 2 allele WW, and the black color, which is recessive, has allele. For you to see black, for you to have black, you will have the black color with two alleles, two of the alleles must be dominant. For you to see black, then the B allele, the black gene is called a 2B gene, 2 capital letter B2. That is 2 alleles for black color. Must be in dominant condition. Any of the 2 must be in dominant condition. For it to be white, any of the white will be dominant condition. And that is why you see 12 white. Anywhere for the sheep, when you cross black sheep and white sheep and black sheep together, any sheep that will show white, the W, any out of the 2 alleles for white color gene, 1 must be in dominant condition. And that's why you will see 12. And black 3. And for you to have black, black must be in dominant. For you to have black must be in dominant condition. You see, when black is in dominant, you see this one is black, and black. 3. Now, for you to have black, B allele will be in dominant condition. And the white color, white allele, the 2 white alleles will be in double recessive condition. Meaning that it will be WW. So WW with 1 dominant gene, dominant B allele beside it, it will give you black. WW with 1 dominant B allele will give you black. And when you have double recessive white allele and double recessive B allele will give you brown. Am I speaking sense? So when you have double recessive white allele, double recessive black allele will give you brown. It will not give you black. For you to have black color, black, 1 out of the 2 alleles of the black will be dominant condition. And the 2 white gene will be in double recessive condition. The 2 white alleles will be in double recessive condition. Why any of the 2 black gene, black allele will be in dominant condition, it will give you black gene, black color. But where you see the white allele is in double recessive condition, the black allele is in double recessive condition, the sheep will be brown. Am I speaking sense? So when the alleles are all in double recessive condition, it will give you brown. Am I speaking sense? The next type of epistasis I'm going to is duplicate dominant epistasis. This duplicate dominant epistasis is a form of epistasis that deviates from simple Mendelian inheritance. And like I said, in double recessive, in dominant epistasis deviates from simple Mendelian inheritance, when you cross black sheep and white sheep together, the phenotypic ratio you have is 12 to 3 to 1, meaning that 12 white sheep, 3 black sheep, and 1 brown sheep. Am I speaking so? That is 12 to 3 to 1. 12 to 3, that is the dominant epistasis. So it will deviate from simple Mendelian ratio and it will become 12 to 3 to 1. Duplicate dominant epistasis deviate from simple Mendelian, but this one is manifested, for example, in a domestic fowl, in feather of shank. If you are very critical, when you see your chicken during Christmas or during other festive season, some chickens will have feather on their shank. Shank is that one they need to give to children to eat, and you'll be sucking the bone marrow, the leg of chicken. I'm sure all of you to eat it when you were small, and you are still eating it now. That leg, it can be white in some chicken, it can be yellow in some chicken. Am I speaking sense? Am I speaking sense? You know, some of them used to have ears. Who have observed that the leg of chicken, you call it shank, used to have ears, some small feathers. If you used to observe it, let me see your hands up. Fantastic, fantastic. Khadija can confirm that. Blikizu can confirm that. Fantastic. Now, on the ear, the feathering of shank, having ears on the shank, the presence of ear and absence of ear on the shank of chicken is controlled by duplicate dominant epistasis, not simple Mendelian inheritance. My speaking sense. My speaking sense. And that one can be seen when you see in where you have F, capital F, 2 capital F, and 2 capital S. You cross it with recessive, double recessive F and double recessive R. Meaning that 2 small letter F and 2 small letter S. That is F for feather shank, F for feather, S for shank. Do you understand? So the gamete there will be segregation. And in this, after crossing, after manifestation, you will have a striking deviation from simple Mendelian ratio of 9 to 3 to 3 to 1 to 15 to 1. And let me explain how duplicate dominant epistasis. Duplicate dominant epistasis give us phenotypic ratio of 15 to 1 ratio. We are talking about feathering of shank in chicken. Feathering of shank in chicken, 15. All of the 15 is feathered shank. All out of the 16 observations, you know, in that hybrid crossing, you will have 15 observations from the gametes. So you have 15 feathered shank and 1 unfeathered. Look at the name of duplicate. Look at the name. Look at the name of the epistasis. Duplicate dominant epistasis for the name. Duplicate dominant epistasis. Meaning that for you to have a feathered shank, the F gene for feathering of the shank, the F for feathering. No, S is for feathering. F is for feathering. You understand? F is for feathering. S is for shank. Shank gene. You understand? So we have 2 allele, 2F allele for feathering, 2S allele for shank. Am I speaking sense? 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 Now, out of when you do Punnett square method crossing, you will have 15 feathered shank and one unfeathered shank. Look at the name, duplicate dominant epistasis, meaning that between the two, you to have feathered shank, the two alleles of F and the two alleles of S, at least one of the two alleles must be in dominant condition. Am I speaking, sir? Should I come over again? Should I come over again? When you have the two alleles, we have two F alleles for feathering and two S alleles for shank. So a feathered shank, when you see a chicken that has feather on its shank, means that the F alleles, the two F alleles and the two S alleles are in dominant condition. And the condition, a major condition to have a chicken with feathered shank is when out of the two alleles of F and two alleles of S, at least one must be in dominant condition. Am I speaking sense? At least one must be in dominant condition. At least one. At least if F is in dominant condition and S is in dominant condition, when you are discussing between this chicken, the feathered chicken and unfeathered feathered shank chicken and unfeathered shank chicken, if you cross 15, the phenotypic ratio, you have 15, at least 15 will be feathered shank. And unfeathered will be, all of them will be in double recessive condition. F will be double recessive, S will be double recessive. Do you know why? Because the name, the name of the place is duplicate, is a duplicate dominant epistasis, meaning that from the two locus, two loci, at least out of the two alleles of F, one must be in dominant condition. And out of the two alleles of S, one must be in dominant condition. Any condition that gives you that will provide you feathered shank. Am I speaking sense? And we only have one unfeathered shank, which are all recessive across all loci. The two alleles of F is recessive. The two alleles of F is recessive. So they are all recessive across all loci. That's why you have, and you have for only one. And that's why you have ratio 15 to 1. Am I speaking sense? Am I speaking sense? Am I speaking sense? Please ask me questions. By the time I finish, I want to go to question and answer section. I want to see a lot of questions so that I'll be able to answer your question. The more you ask, the more you ask me questions, the more I will have latitude to answer many of your questions and brighten your understanding. Am I speaking sense? Am I speaking sense? And the last one we will be speaking to is a duplicate, is complementary epistasis, which is called duplicate recessive epistasis. The one I just finished is duplicate dominant. We have, I've spoken to recessive epistasis, dominant epistasis. I've spoken to duplicate dominant epistasis. Now I'm not speaking to duplicate recessive epistasis. Duplicate recessive epistasis is also called complementary epistasis. Example of this is found in the color code. Let me say, example of this is found in rabbit color codes. Color code type of rabbit, normal color code, and Rex color code. The Rex color code, REX, is short-eared coat. The hair is short, it's not as long as normal one. So a Rex color code and a normal color code. Am I speaking sense? Am I speaking sense? Am I speaking sense? Now, then you cross a normal color code with a Rex color code, and you now see a demonstration. You see a demonstration of duplicate epistasis or complementary epistasis. And when it says complementary, from as the name sounds, something is complementing, is adding value to another, is making another one, is storing up another one's effect. And in this kind of epistasis, we have ratio 9:7. Ratio 9:7, a striking deviation from simple Mendelian inheritance. 9 is normal. It's normal color code of rabbit. While the red color with short hair is 7. And the condition that will make it to have this is because it's complementary. Listen, it's complementary. Meaning that we have reds for you to see a normal color. For you to see a normal color, you will have reds is, let me say, you can say R1, R2 is for normal color. And you see recessive, double R is for normal color. You know, 2 colors are involved. Code type, normal and rex. Normal and rex. The normal, the 2 allele for normal is capital R. The 2 allele for rex is recessive R. Small r, 2 small letter R, that is recessive. When the color is normal, is 2 capital R. When the color is recessive, is 2 small letter R. So when the color when for in this one when you know these are two color code short hair, we have rex is short haired coat, while normal color is normal color coat. I'm still speaking to complementary epistasis or duplicate epistasis. Don't forget, when you have two dominant allele, that is homozygous dominant. When you have 2 recessive allele, 2 small letter R is heterozygous. So when you have 2 capital alleles, 2 capital letters for 2 alleles is a dominant homozygous. 2 small letters for 2 alleles is called recessive homozygous. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Now, this ear is normal, long and short. The rest color. Now, for you, and I said the phenotypic ratio for this one is 9:7. The normal type, you will have capital R with 2 recessive R beside the 2 recessive R. 2 recessive R. 2 recessive R. Am I speaking, sir? So this is normal. 2 dominant R and 2 recessive R. 2 dominant R and 2 recessive R. So for you to have a normal rabbit color, the dominant R, let me say, R1, capital R1, capital R1, R2, and let me say R1, small recessive, 2 recessive, R1, R2, let me say R3, 2 alleles. For you to have a normal color, At least the R1 must be in dominant condition, and dominant R, capital letter R, which is dominant R, at least one of the dominant R must be in one of the R alleles must be in a dominant condition, and one of the R3 allele must be in dominant condition. At least, don't forget, I said the color, the normal color is dominant, two dominant R1, and two recessive R, small letter R, R3. That's all. The Rex is the Rex color. The Rex color is the recessive. Is the recessive. Am I speaking sense? But most speaking sense, storm down for me. So let me explain myself. But most speaking sense, storm down for me. That is fine. So I said the normal color and Rex color. For you to have a normal color, you must have at least one dominant R. At least one dominant R in one locus for you to have normal color. And you have one dominant R1 and another dominant R3 for you to have a normal color, complementary. At the two loci, you must have at least one dominant normal color, non-dominant R, R1 and R3. That's what will give you normal color, at least. We have R1, R capital letter, how dominant R1, 1, 2. You know, the two alleles. And recessive, R3. Now, for you to have a dominant normal color, let me say normal color, R1 will be at least dominant, R3 will be at least dominant. Complementary. When I say what complementary? One is helping each other. So you must have a dominant R allele, R1 and R3 allele for you to have a normal color. And when you have Rex, so if you count that one, you see 9. You're going to have 9. You're going to have 9. So then you will have 7 for Rex. How do you get Rex? These are the conditions that will make you to get Rex. When all the recessive have, when all R are in recessive condition, you get a Rex color. When all R are in recessive condition, mean that all the allele, all the 4 allele are R, you will get a Rex color. When you have 2 recessive R, when you have 3 recessive R and 1 dominant R, you will have Rex color. If you have 2 recessive R, 2 recessive R1, and 2 recessive R3, you will have Rex. You will have Rex. If you only have 1 dominant R, dominant R1, you will have Rex. Meaning that you must have at least 2 dominant R1 and R3 for you to have a normal color. Don't forget, I said to you, the allele controlling this color is this. Normal color, you have R1, dominant R1, and 2 recessive R3. That is normal coat color, rabbit. We are crossing it with 2 recessive R1, and 2 dominant R3. So between two dominant R1 and recessive R3, We have R3 in recessive condition 2. We have R1 in recessive in dominant condition 2. So for you to have a normal color, out of the two dominant R1 allele, at least one must be in dominant condition. Out of the two recessive R3 allele, at least one must be in dominant condition. Then you will have normal color, and it will be 7, and the rest will be Rex. Am I speaking sense? 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 Having said this, these are the explanations. I'm going to send you the recorded version of this class, and I'll send you the summary notes, the note of this class. I'm going to send it to you probably tomorrow, so that you really understand. Later, we'll upload the entire notes for you to be able to download. Let me see questions. I'm sure you might have got a lot of questions for me now. No questions for me. No questions for me. No questions for me. Any question? Any questions? Please ask me questions so that I can answer some of those things you don't understand. Questions? I'm saying no questions. Please ask me questions, please. Please ask me questions. Please ask me questions. Ask me questions. Should I do a summary, a run over of what I've taught? Should I do a run over of what I've taught? Okay, fantastic. I have spoken to you. I did a summary of what I taught in previous class, and that led me into this class. We are all talking about gene actions. Simple Mendelian inheritance is a form of gene action. It's a form of dominant gene action. Simple Mendelian inheritance is a form of dominant gene action. And majority of simple characters are governed by simple Mendelian inheritance. The obeyed law of Mendelian law of independent, Mendelian law of inheritance. The obeyed first law of Mendelian law of inheritance, which is law of segregation, and second law of Mendelian inheritance, which is law of independent assortment. Now, but there are some form of inheritance, some form of genetic actions that disobey simple Mendelian inheritance. And one of them is crossing over. One of them is gene linkage. One of them is epigenetics. And one of them is gene modification, epigenetics, epistasis. And I told crossing over is when two adjacent chromosomes lying, crossing over occurs between non-sister chromatids. It occurs between non-sister chromatids, meaning that two chromosomes are adjacently lying side by side, and they exchange chromosomal part. The point at which they exchange chromosomal part is called chiasma. Chiasma. The point of exchanging chromosomal part called chiasma. When there are many, it's called multiple manifestation of chiasma. It's called chiasmata. That is a multiple. When the chromosome is long,
when they list side by side, there is possibility that they can exchange chromosomal part, they can cause each other at multiple points of chiasma. So multiple manifestation of chiasma is called chiasmata. So, and the major advantage of crossing over is to create genetic diversity. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. So, but crossing over occurs between two non-sister chromatids. Non-sister chromatids. Why linkage? When gene linkage occurs between two sister chromatids, between the same sister chromatids. What crossing over occurs between non-sister chromatids? We have two chromatids that make a chromosome. Two chromatids make a chromosome. So crossing over occurs between non-sister chromatids, while gene linkage occurs between the same sister chromatids. Same homologous chromosome. Same things within the same homolog. So such that the gene did not segregate as the gene on the same chromosome pair, on the same sister chromatid, on the same chromosome pair, refuse to segregate due to non-disjunction. They refuse to segregate. They refuse to segregate. Am I speaking sense? Am I speaking sense? They refuse to do what? They refuse to segregate. And they are now being inherited together. They refuse to segregate. They are now inherited together. Am I speaking sense? Am I speaking sense? So when they refuse to segregate, they are inherited together. That is gene linkage. An example of gene linkage is coupling and repulsion. Coupling and repulsion. Coupling when a dominant allele and recessive allele stick together and they are inherited together. That is coupling. And repulsion is when two recessive alleles or two dominant alleles are inherited together. So it occurs with linkage occurs within the same homologous chromosome pair, within the same chromosome, between two sister chromatids. That difference between linkage and crossing over. Crossing over happens between non-sister chromatids, while linkage occurs between two sister chromatids, between the same homolog, between homologous pair. You know, we have two chromosomes governing the same. You know, we have two alleles. I'm speaking say two alleles governing the same character. Two alleles control the same character, isn't it? Am I speaking, sir? So the individual allele is residing on two chromosomes. Instead of the two chromosomes to segregate, they refuse to segregate. The alleles on them, the genes on them are inherited together. That is gene linkage. Am I speaking sense? Am I speaking sense? Am I speaking sense? And I went further to speak to epistasis, when a gene at one locus masks the expression of other gene at another locus. Interallelic interaction. And I speak to 4 different types of epistasis. Number 1, recessive epistasis, which which is demonstrated in the crossing between color of mice, agouti mice, and black and albino mice. Agouti mice and albino mice. That is rats. When you cross agouti, you form a black and white color rat with albino, you have a phenotypic ratio that is of striking deviation from simple Mendelian ratio. 9-3-3 to 1, you now have 9 agouti, 3 black, and 4 albino. And when you have albino, the black color will be in double recessive condition, recessive epistasis. The color, the black, the agouti color, which is the agouti color, for you to have agouti color, C for color, for black color, must be in dominant condition, and A, albino gene for color, will be in dominant condition. Then you have agouti. You have agouti. When C, that codes for color, that codes for black color, is in dominant condition, and A, that codes for albino color, is in dominant condition. So when C is in dominant condition, A is in dominant condition, you will have agouti. You know, no, albino rat is white. Albino rat is white. So meaning that when you have black color mixed with white color, you're going to have gray, which is an agouti. Am I speaking sense? 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 So then, that is a good recessive epistasis will give you ratio 9-3-4. Then the second is dominant epistasis, which is demonstrated in the color code of color pattern of fleece of sheep, white sheep and black sheep. And when you cross that, the dominant epistasis will give you 12 ratio. The phenotypic epistatic ratio will not be 12-3-1. 12 white sheep, 3 black sheep, and 1 brown sheep. We have dominant we have duplicate dominant epistasis. Duplicate dominant epistasis, which between the feathered shank and unfeathered shank in chicken. Then you will have 15 ratio 15-1. That's the epistatic ratio. Because for you to have a feathered shank, the hair allele and S allele, F allele and S allele must be in dominant condition. F allele, and S allele at least must be dominant, in dominant condition. That is dominant duplicate epistasis. Then we go to the last one, epistasis, complementary epistasis, which gives you 9-7 epistatic ratio. So I have come to the end of this class. Is it interesting? Is it interesting? Fantastic. I'm seeing Ibrahim, I'm seeing Billy Kisu, I'm seeing a lot of people. Fantastic. So I'm going to send, at the end of this class, I'm going to send the audio, the recorded version. Miriam, I'm seeing you. Miriam Yusuf, I'm seeing you. I'm seeing Manasseh too. I'm seeing Blikisu. I'm seeing Adeja. I'm seeing Perpetual. I'm seeing Yaya. I'm seeing Barakat. Barakat is fantastic. Barakat is fantastic. Barakat is fantastic. So I'll send the recorded version to Barakat. Barakat will upload. We'll share with you the summary note, the entire notes in a word version. I'm going to send to you. So by the time you read this, you will be able to have a better understanding. At the end of the day, the PowerPoint version will be uploaded so you'll be able to download it from your portal. Am I speaking sense? Am I speaking sense? Am I speaking sense? And nobody's asking any questions. So I want to believe you have a good understanding of what I've been teaching you. Isn't this class interesting? Isn't this class interesting? Isn't it interesting? If you're interested, let me see a lot of thumbs up. Thank you very much for giving me your time. Thank you very much for giving me your time. The next class I'm going to have with you, I will let you know. And then we'll have a test, we're going to have an online test too. I will let you know. So thank you for your time and have a beautiful night rest. Thank you. Bye-bye.